Good day, everyone. My name is Celine Eliza Abler. I will be discussing bacterial transformation for advanced genetics subject. This topic will discuss the history of the discovery of the DNA, the structure of the DNA, doubt on the capacity of the DNA as a vehicle of heredity, pursuit for the molecule responsible for heredity, process of bacterial transformation, chemical fractionation of the transforming particle. Let's start with a discussion of the history of the discovery of the DNA. At the dawn of the 20th century, scientists have not yet proven that the DNA is the genetic material capable of carrying the heritable traits from parents to offspring. In 1869, Friedrich Miescher isolated the first crude extract of DNA. Upon the discovery of the nuclein after purifying DNA, it was identified that DNA contains four distinct chemical building blocks arranged in a polymer, a long chain of repeating units. So what is the structure of the DNA? DNA stands for deoxyribonucleic acid. The term deoxy refers to the sugar or the pentose sugar in deoxyribonucleic acid, which is deoxyribose. Deoxyribose is a pentose sugar because it has five sides. There are five carbons that makes up the sugar. So in DNA, only the deoxyribose is used as a sugar, whereas in RNA, it is the ribose sugar. Also, upon the discovery of the deoxyribonucleic acid, the term nucleic refers to where the deoxyribonucleic acid was discovered. So it was in the nuclein or inside the nucleus where the DNA was found. So the DNA was already isolated. When it was isolated, it was found to be part of the nucleus of a cell. So it is inside the nucleus of the cell. So the DNA was actually found using the Fulgen reaction in 1923. It was during the pursuit of the scientists when they were looking for the factor for the heredity, which is they were debating whether it was between protein or the DNA. So one of the scientists actually performed a reaction to determine where the DNA resides. So the staining technique was called Fulgen reaction, which uses a Schiff reagent to stain the chromosomes red and led to the discovery that the DNA resides almost exclusively in the chromosomes, which are normally found inside the nucleus. So these are the examples of cells which were stained using the Fulgen reaction, using the Schiff reagent for the Fulgen reaction. So these are the cells stained to reveal the location of the DNA in the nucleus. So we notice that the darkening or the reddening of the portions refer to the location of where the chromosomes or the DNA would be found. So these are the sample of plates where cells were stained using the Fulgen reaction. Another reason why the DNA was named as such or as deoxyribonucleic acid was that it came from the term acid. Okay, the term acid actually refers to the acid which Miescher found between the reactions between the nitrogen and the phosphorus in the skeleton of the DNA. So he found that one of the skeleton actually contains phosphoric acid, which makes up the phosphate group in the skeleton of the DNA. So this is the structure of the DNA and the chemicals that compose it. So the reason why we need to understand first the structure of the DNA, it's because the DNA is a very important factor for the heredity of the transformation for the bacteria, which we will be discussing later. So the bacteria actually has the same structure for our DNA. It also has region where it can store the DNA. We call it the nucleoid region. And the DNA that and the chemicals that compose the DNA are actually still the same. So the DNA is actually very precious for the scientist who wants to prove that it is the hereditary factor. So the understanding of the structure of the DNA actually led to the development or the experiment that proved that DNA is the one that is used for the transformation of the bacteria and therefore can explain the heredity of the organisms or the heredity of the genes for all organisms. So before DNA became the hereditary factor for all organisms, there was a scientific inquiry about whether it has the capacity to become the vehicle of heredity. So we will be discussing this now. So before the advent of DNA as the hereditary factor, scientists had doubted the capacity of the DNA as the vehicle factor for the hereditary of traits. So they thought that it was too simple to specify the complexity of the genes because the genes, they really think that all genes are equally complex regardless of whether it was a very large animal or organism or a very little or tiny microscopic organism. So for this pursuit, for the molecule responsible for the heredity of genes, so this was a lot of scientists in the scientific community was actually skeptical whether it was the DNA. So they really thought that it was the proteins because proteins were always responsible for a lot of things back then and even now, but they really thought that it was proteins, not the genes or not the DNA that was responsible for heredity. So let's start with whether or not it was really DNA and what are the experiments that were responsible for the discovery of this and this DNA as the proof for the heredity of genes. So for the process of bacterial transformation, we will first discuss what is bacterial chromosome versus the human chromosome. So this is the bacterial chromosome on the left, which is a little bit circular. So most of the scientists actually describe it as a circular chromosome when it is arranged and ready for cellular division, which is binary fission for bacteria. And the human chromosome is arranged as a cross or an X for when it is getting ready for cellular division, whether it is mitosis or meiosis. So for human chromosome, a lot of proteins are actually responsible for the structure of the entire genetic material. There are histones that composes the human chromosome, whereas for the bacterial chromosome, it is really simple compared to the human chromosome. So the process of looking out whether or not DNA was the factor that is responsible for heredity was set out by Frederick Griffith. He was very known for the process of bacterial transformation, which he performed in 1923. So there were several studies before him and that were conducted to support the theory that DNA was the one that is responsible and can carry the genetic information from one generation to another. So during his experimentation, he found out and identified the two forms of Streptococcus pneumoniae, which are the wild type, which is smooth under the microscope, and the mutated type, which is rough under the microscope. So these are the rough colony of the Streptococcus pneumoniae. So we notice on the left, we notice that it is rough because it has no capsule, which is used by the bacteria to protect itself from the immune response or immune attack of the immune response of the host where it colonizes. And another one is the smooth colony. The smooth colony is um, the one that has the capsule, the polysaccharide capsule actually allows the bacteria to prevent detection or evade detection by the host's immune system. So the one that is really virulent and the one that causes disease or death is the one that forms a smooth colony, which is the wild type or the S form, and the one that is the rough colony or the mutated type or the rough form is the one that is virulent and cannot cause disease and can be destroyed by the host's immune system. So this is Griffith's experiment, and the discovery of the transforming principle for the DNA, which is responsible for the genetic or the hereditary factor. So this experiment actually has shown that a dead S strain can actually transfer its genetic material into a living R strain, which is on setup 4. So we should also note that since the polysaccharide capsule protects the bacteria from the immune response of an animal, the S form is considered as virulent. So the one that is smooth, the one with the polysaccharide capsule, it is the one that is virulent and capable of killing most of the laboratory animals that are exposed to them. So in contrast, because of mutation, the R form actually fails to cause infection for those animals that they were introduced into. So that's why the living R strain, which is non-virulent, can allow a mouse to live, which is on setup 2. So for the setup 1, it is expected that the living S strain would cause a mouse to die. But for the living R strain, because it has no polysaccharide capsule and is not able to evade the host's immune system, so it will be destroyed and will allow the mouse to live. Now here comes the coolest part of his famous experiment, which he published in 1928. So he found that when he injected a living R strain, which means that this R strain has no polysaccharide capsule, and he allowed it to be alive, and that he injected it into a mouse, and he also injected a heat-killed S strain, which is the one that is virulent, into a laboratory mice. So the mouse will live only if it's only the dead S strain that is injected into the mouse. But if you combine or mix the living R strain and the dead S strain into the mouse, when injected, the mouse is um, is found out to be dead, and it is found out that the S strain is capable of producing infection, and that he found out also that the strains recovered from the blood of the dead mice were all living S forms. So meaning that there was a transformation that happened within the organism. The term transformation in genetics is a phenomenon that refers to that the genetic characteristics of an organism was already changed. So initially you were expecting that it is only the living R strain that will be carried into the next generation of the bacteria. However, the S strain, although it is already dead, was able to transfer its genetic factor or genetic material into the living R strain, and um, was capable of transforming it to carry its genetic material into the next generation of the bacterial colony that it will be dividing into. So this transformation, which Griffith found out through this experiment, appears to be permanent and mostly genetic or heritable, which is carried out to the next generation again. And as all future and succeeding generations of the bacteria grown in culture were all S forms or smooth forms, or all that carry the capsule or polysaccharide capsule that protects it or let it evade the host's immune system. So let's now conclude or go to the experiment that actually solidified the evidence of DNA as the transforming principle or transforming particle for the heredity of genes or heredity of traits. So one of the experiments was called chemical fractionation. So it was actually performed by Oswald Avery. His life's goal was actually to try to identify whether or not the heritable substance in the bacterial extract, which Griffith was able to perform in 1928, induces the transformation of harmless R bacteria into pathogenic or disease-causing S bacteria. So he was looking for the transforming principles, which is really what was really responsible for the transformation in the bacteria experiment done by Griffith. So he was aided by his friends or colleagues, which were MacLeod and McCarthy. So they were looking for the DNA or proving the DNA as the substance for genetic inheritance. So they were against the um, hypothesis of Mirsky, which thought that or which posits that the protein is the substance of genetic inheritance. So let's remember again that in Griffith's experiment, the hypothesis was that there is a material in dead bacterial cells that can transform living bacterial cells. So this was the basis for Avery's pursuit for the heritable factors. So the conclusion in this experiment, in Griffith's experiment, is that there must be a chemical substance of one cell that is genetically transforming another cell. So this was the one that Avery used for his experiment on chemical fractionation of the transforming particle. So this is Avery's experiment for the transformation experiment. So his hypothesis was initially that genetic material of the cell can either be a protein or a nucleic acid. So it can be either a protein, a nucleic acid, which is DNA or RNA. So his goal was to try to identify the heritable substance in the bacterial extract, which can induce the transformation of the harmless R bacteria into pathogenic disease-causing S bacteria. So he called the substance as the transforming principle. So this experiment was actually to prove whether or not it was a DNA or RNA or the nucleic acid or the protein that was responsible for the transforming particle. So for this experiment, he used heat-killed S cells and removed the lipids and sugars from the solution of the heat-killed S cells. So he was only the proteins, RNA and DNA remain. So in order to identify whether it was the RNA, DNA, or the protein that was able to transform the added R strain of cells. So he will be adding enzymes to try to dissolve or metabolize or degrade the material that is the target for this experiment. So he added proteinase to dissolve the protein to look whether or not it will be the protein that will be responsible. He added RNAs to look whether or not it was the RNA that was the reason for the transforming principle, and he added DNAs, the one that can degrade DNA, to look whether or not it was the DNA that was responsible for the transforming principle. So when he added in the R cells, when there was no protein, S cells still reappear. So when S cells still reappear, that means there was still transformation that still happened in the culture containing the living R cells. Another thing is that when he added RNAs and therefore there are no RNA inside it, still S cells appear. So that means it was still able to transform the bacterial genetic factor. But when he added DNAs, he found that there are no S cells that actually that actually proliferated. Instead, they only found R cells, living R cells, and that there are no presence of virulent S cells in the culture. So therefore, as his conclusion, the transformation requires DNA. So therefore, it will be noted that it is the genetic material of the cell, the one that is responsible for the transformation that happened in Griffith's experiment on bacterial transformation. So those enzymes that he used to identify whether or not it was DNA, RNA, or it was protein that was responsible for the transforming factor, were able to degrade protein, RNA, and DNA, and it was able to prove that that only DNA is the genetic material of the cell. So when they subjected the purified substance to an enzyme that can degrade the DNA, then they have seen that it has completely destroyed the activity of the transforming principle. So that was it. That was bacterial transformation. Bacterial transformation actually starts with the discussion of whether or not it was really DNA that was the one that is responsible for the heredity of genes or the heredity of traits, and the discussion of what was in the DNA, why it was really important to discover whether or not it was the heritable factor, and the processes of bacterial transformation, which was conducted by Griffith in 1923 and was published in 1928, and the one that is the one that actually purified the form of the DNA in Avery's experiment, which is published in 1944. So it was Avery that actually proved Griffith's bacterial transformation experiment. He proved that DNA was the one that is responsible for the genetic transformation in that happened in the bacteria, which is Streptococcus pneumoniae, between S form, the smooth form, and the rough form or the R form. So thank you for learning this lesson with me. So the reference for this topic can be found in Hartwell's Genetics from Genes to Genomes, 4th edition. So thank you, and good day everyone, and have a blessed day.